Germination is the process by which a plant grows from a seed into a seedling. Seeds remain dormant until conditions are favorable for germination. All seeds need water, oxygen, and optimal temperature to germinate. When a seed is exposed to the proper conditions, water and oxygen are taken in through the seed coat. The embryo's cells start to enlarge. Then, the seed coat breaks open and the root emerges first, followed by the shoot that contains the leaves and stem. Sunlight supports the germination process by warming the soil. Although uncommon, some seeds need direct contact with sunlight to germinate. Once leaves have sprouted, sunlight is the primary energy source for plant growth through the process of photosynthesis. Phases of growth in plants. There are 3 phases of growth: meristematic or formative, elongation, and maturation. We can understand this better by looking at a seed. We already know that the tips of roots and shoots exhibit continuous growth and hence are meristematic. The cells in this region are rich in protoplasm and have large nuclei. The cells next to this region represent the elongation growth phase. In this region, there is the formation of new cell walls, vacuoles etc. The region next to the elongation phase represents the maturation phase in which the cells attain their maximal size. Growth rates. The number of cells in organisms increases in a number of ways. The increased growth per unit time is termed the growth rate. This can be expressed mathematically. There are 2 types of growth rates: arithmetic and geometric. In arithmetic growth rate, out of the 2 daughter cells produced by the mitotic division of a cell, only one daughter cell continues to divide while the other differentiates and matures. Absolute growth rate is total growth per unit time. In the given representation, the absolute growth rate for both leaves is 5 square centimeters per given time. Relative growth is the rate of growth with respect to the initial size. Crop growth rate (CGR) expresses the gain in dry matter production of the crop per unit land area per unit time and is expressed as gram per meter square per day (gmm2 day 1). Some of the elements essential for the growth of plants are oxygen, water, and nutrients. Water enters the cells and causes turgidity. This results in the enlargement of the cell. Water also provides a medium for the activities of enzymes. The metabolic energy required for various activities is obtained from oxygen and sometimes nutrients. Optimum temperature, light, and gravity are also required for the proper growth and development of the plant. Air. Plants need air to breathe and grow well, just like we do. Plants take in CO2, carbon dioxide, and then photosynthesis takes place to convert the CO2 into food. Differentiation occurs in plants when cells from the root apical and shoot apical meristems, as well as the cambium, differentiate and mature to execute specialized roles. Living plant cells lose their ability to divide once they have been differentiated. However, under some circumstances, this power to divide further can be recovered. Dedifferentiation is the process by which mature cells reverse their differentiated state and acquire pluripotency. Redifferentiation is the process by which dedifferentiated cells lose their ability to divide and become specialized to perform a role by converting into a part of the permanent tissue. The main distinction between dedifferentiation and redifferentiation is this. The stages that plants go through are from seed to sprout, then through vegetative, budding, flowering, and ripening stages. The correct sequence of the development process in a plant cell is cell division, elongation, differentiation, and maturation. The development process of a cell refers to the changes that occur in the cell from cell division to maturation. The source of all plant cells, i.e. xylem, phloem, pith, etc., is apical and shoots meristem. At the meristem, cell division occurs, and after division, daughter cells further undergo plasmatic growth and differentiation, further elongation and maturation. Finally, the plant cell becomes old and dead senescence. Plant growth regulators are the chemical substances which govern all the factors of development and growth within plants. Some other names used to refer to it are phytohormones and plant growth hormones. Phytohormones are organic compounds which are either synthesized in laboratories or produced naturally within the plants. They profoundly control and modify the physiological processes like the growth, development, and movement of plants. Based on their actions, Plant growth regulators are broadly classified into two major groups. Plant growth promoters, plant growth inhibitors. Auxins, gibberellins, and cytokinins are grouped into plant growth promoters, while abscisic acid and ethylene are grouped into plant growth inhibitors. Ethylene can be grouped either into the promoters or into the plant inhibitors. Auxin is mostly produced in the apical meristem of shoots, young leaves and seeds. Movement of auxin is unidirectional or polar, it moves downwards from its site of production. Polar transport results in an auxin concentration gradient, which stimulates specific responses. Auxin-specific transport proteins in the plasma membrane control the movement of auxin out of the cell. Plant hormones act by signal transduction, eliciting more than one cellular responses. Auxin binds to enzyme-linked receptors, which promotes catalysis of a reaction. When auxin binds to a receptor, it initiates binding of a repressor protein for certain genes, auxin response gene to ubiquitin, resulting in degradation of repressor protein, and the transcription of auxin response genes progresses, leading to cellular growth and development. Gibberellins gas are plant hormones that regulate various developmental processes, including stem elongation, germination, dormancy, flowering, flower development, and leaf and fruit senescence. Gas are one of the longest known classes of plant hormone. It is thought that the selective breeding, albeit unconscious, of crop strains that were deficient in GA synthesis was one of the key drivers of the Green Revolution in the 1960s, a revolution that is credited to have saved over 1 billion lives worldwide. Cytokinins are plant-specific chemical messengers, hormones that play a central role in the regulation of the plant cell cycle and numerous developmental processes. Cytokinins were discovered by F. Skoog, C. Miller, and co-workers during the 1950s as factors that promote cell division, cytokinesis. Cytokinins are a group of plant growth regulators which are primarily involved in performing cell division in plant roots, shoot system. This hormone helps in promoting the cell's growth, development, differentiation, affecting apical dominance and delay in leaf senescence. Ethylene is the first identified plant hormone known to regulate numerous processes in plant growth, development, and response to biotic and abiotic stresses. Ethylene is best known for its effect on fruit ripening and organ abscission, and thus has great commercial importance in agriculture. As a gaseous hormone, ethylene can freely diffuse across membranes and is thought to be synthesized at or near its site of action, which is different from other plant hormones. Abscisic acid, Aber, is a plant hormone. Aber functions in many plant developmental processes, including seed and bud dormancy, the control of organ size and stomatal closure. It is especially important for plants in the response to environmental stresses, including drought, soil salinity, cold tolerance, freezing tolerance, heat stress, and heavy metal ion tolerance. Aber was originally believed to be involved in abscission, which is how it received its name. This is now known to be the case only in a small number of plants. Aber-mediated signaling also plays an important part in plant responses to environmental stress and plant pathogens. The plant genes for Aber biosynthesis and sequence of the pathway have been elucidated. Aber is also produced by some plant pathogenic fungi via a biosynthetic route different from Aber biosynthesis in plants. Photoperiodism is the physiological reaction of organisms to the length of night or a dark period. It occurs in plants and animals. Plant photoperiodism can also be defined as the developmental responses of plants to the relative lengths of light and dark periods. They are classified under three groups according to the photoperiods: short day plants, long day plants, and day neutral plants. Vernalization is the induction of a plant's flowering process by exposure to the prolonged cold of winter, or by an artificial equivalent. After vernalization, plants have acquired the ability to flower, but they may require additional seasonal cues or weeks of growth before they will actually do so. Some examples include beets, onions, winter wheat, cabbage, and turnips. In order to produce flowers and seeds, these plants have to go through a process called vernalization. Vernalization simply means that the plant has to experience a period of cold before it can produce flowers. Photoperiodism is the physiological reaction of organisms to the length of night or a dark period. It occurs in plants and animals. Plant photoperiodism can also be defined as the developmental responses of plants to the relative lengths of light and dark periods. They are classified under three groups according to the photoperiods: short day plants, long day plants, and day neutral plants. Photoperiodism is the physiological reaction of organisms to the length of night,